Welcome to Hormone Cyclopedia. Before we begin this discussion about cancer as a metabolic disease, I need to share an important disclaimer. The information presented in this video is for educational purposes only and should not be considered medical advice. While we discuss emerging research about cancer's metabolic aspects, this information does not replace conventional cancer treatments or professional medical care. Every cancer case is unique, and treatment decisions should be made in consultation with qualified healthcare professionals who are familiar with your specific medical history and condition. The concepts discussed here about cancer metabolism are based on current research, but are still evolving, and more studies are needed to fully understand their clinical applications. If you have cancer or suspect you might have cancer, please seek immediate medical attention from qualified healthcare providers. Do not attempt to self-diagnose or self-treat based on this information. I'm Dr. Richard Nkwenti, and while I'm a medical professional, I'm not your personal physician. Always consult with your healthcare team about your specific situation and treatment options. Now let's explore this fascinating topic about understanding cancer as a metabolic disease. Exploring the relationship between cancer and metabolism. The intricate interplay of cancer cells and metabolism. Metabolism is a fundamental process in all living cells, providing the energy and building blocks necessary for cell growth, proliferation, and maintenance. In cancer cells, metabolism undergoes significant alterations to support their rapid growth and survival. These changes in metabolism are not just a consequence of uncontrolled cell division, but also actively contribute to tumor genesis and progression. Metabolic adaptations as a survival strategy for cancer cells. Cancer cells exhibit metabolic flexibility, allowing them to adapt to the dynamic microenvironment within tumors. This adaptability enables cancer cells to thrive under conditions of nutrient scarcity or oxygen deprivation commonly found in solid tumors. By rewiring their metabolic pathways, cancer cells can sustain high rates of proliferation and evade cell death signals, ultimately promoting tumor growth and metastasis. Understanding how altered metabolism contributes to cancer development. The role of dysregulated metabolic pathways in tumor genesis. In cancer development, genetic mutations often drive aberrant activation of specific metabolic pathways that provide a growth advantage to transformed cells. For example, oncogenes such as MI can promote increased glucose uptake and utilization by upregulating glycolysis, while loss of tumor suppressor genes like p53 can dysregulate mitochondrial function and enhance nutrient scavenging in cancer cells. Mechanisms linking altered metabolism to tumor initiation and progression. Metabolic reprogramming in cancer cells not only fuels their uncontrolled proliferation, but also influences other hallmarks of cancer such as immune evasion, angiogenesis, and resistance to therapy. By modulating key metabolic pathways involved in biosynthesis, redox balance, and signaling cascades, cancer cells create an environment conducive to their survival and expansion within the host organism. The origins of investigating cancer as a metabolic disease. Historical perspectives on the concept of cancer metabolism. The idea that altered metabolism plays a crucial role in cancer dates back to the early 20th century, when Otto Warburg observed increased glucose consumption in tumor tissues compared to normal tissues, a phenomenon known as the Warburg effect. This pioneering work laid the foundation for studying how changes in cellular metabolism contribute to malignant transformation and provided insights into potential therapeutic strategies targeting metabolic vulnerabilities in cancer. Evolving paradigms, from genetic-centric to metabolism-centric views of cancer. In recent years, advances in metabolomics technologies have allowed researchers to comprehensively profile the metabolic landscapes of different cancers, revealing unique metabolic signatures associated with specific tumor types or molecular subtypes. This shift towards understanding cancer as a metabolic disease has opened new avenues for precision medicine approaches that target the metabolic dependencies of individual tumors based on their distinct metabolic profiles. Glycolysis. Glycolysis is a key metabolic pathway that is often dysregulated in cancer cells. Cancer cells tend to rely heavily on glycolysis for energy production, even in the presence of oxygen, aerobic glycolysis, a phenomenon known as the Warburg effect. This shift towards glycolysis allows cancer cells to produce ATP and other metabolites needed for rapid cell proliferation. Targeting glycolysis has emerged as a potential therapeutic strategy for cancer treatment. Key points. Cancer cells often exhibit elevated rates of glycolysis compared to normal cells. The Warburg effect describes the preference of cancer cells for aerobic glycolysis. Inhibiting enzymes involved in glycolysis may be a promising approach for cancer therapy. Pentose phosphate pathway, PPP. The pentose phosphate pathway, PPP, is another metabolic pathway that plays a crucial role in providing cancer cells with building blocks for nucleic acid synthesis and redox balance. Dysregulation of the PPP can lead to increased production of NADPH, which is essential for maintaining cellular redox homeostasis and supporting rapid cell growth. Targeting the PPP has been explored as a potential strategy to selectively inhibit cancer cell proliferation while sparing normal cells. Key points. The PPP generates NADPH, which is important for antioxidant defense and biosynthetic processes in cancer cells. Increased flux through the PPP can support nucleotide synthesis and promote tumor growth. Inhibiting key enzymes in the PPP could disrupt redox balance and inhibit cancer cell proliferation. Effects of nutrient deprivation on cancer cell metabolism. When cancer cells are deprived of essential nutrients such as glucose or amino acids, their metabolism undergoes significant changes to adapt to the new environment. One common response is the upregulation of alternative metabolic pathways, such as fatty acid oxidation or autophagy, to generate energy and sustain cell growth. These adaptations allow cancer cells to survive under nutrient-deprived conditions and continue proliferating. Key metabolic changes in response to nutrient deprivation: increased reliance on fatty acid oxidation for energy production, activation of autophagy to recycle cellular components and maintain metabolic homeostasis, downregulation of glycolysis and other nutrient-dependent pathways, impact of excess nutrient availability on cancer cell metabolism. In contrast, excess availability of nutrients such as glucose or glutamine can also have profound effects on cancer cell metabolism. High levels of these nutrients can promote rapid cell proliferation and tumor growth by fueling energy production and biosynthetic pathways. This metabolic reprogramming is often driven by oncogenic signaling pathways that stimulate nutrient uptake and utilization in cancer cells. Main consequences of excess nutrient availability: enhanced glycolysis and pentose phosphate pathway activity for increased ATP and nucleotide synthesis, elevated glutaminolysis to support anaplerosis and redox balance in rapidly dividing cancer cells, promotion of de novo lipogenesis for membrane biogenesis and cell growth. Targeting glycolysis. Glycolysis is a fundamental metabolic pathway that cancer cells often rely on for energy production. By targeting key enzymes involved in glycolysis, such as hexokinase and pyruvate kinase, researchers have been able to disrupt the ability of cancer cells to generate ATP through this pathway. Inhibitors of glycolysis have shown promise in preclinical studies, and some have even progressed to clinical trials for various types of cancer. Examples of successes: the drug 2-deoxyglucose 2-DG inhibits hexokinase, a key enzyme in glycolysis, and has shown efficacy in preclinical models of breast cancer and glioblastoma. Aerobic glycolysis inhibitors like linidamine have demonstrated anti-tumor effects in multiple cancer types by disrupting the Warburg effect, a hallmark of cancer metabolism. Targeting fatty acid synthesis. Cancer cells also exhibit increased de novo fatty acid synthesis to support their rapid growth and proliferation. Inhibiting enzymes involved in fatty acid synthesis, such as acetyl-CoA carboxylase (ACC) and fatty acid synthase (ASN), has emerged as a promising strategy for cancer treatment. Several small molecule inhibitors targeting these enzymes are currently under investigation for their anti-cancer properties. Potential therapeutic targets. ACC inhibitors like sorafin A have shown efficacy in preclinical models of ovarian cancer by blocking lipid biosynthesis and inducing cell death. FASN inhibitors like orlistat have demonstrated anti-tumor effects in prostate cancer by reducing lipid accumulation and promoting apoptosis in cancer cells. Mitochondrial dysfunction in cancer cells. One of the key aspects of cancer cell metabolism is the dysfunction of mitochondria. Mitochondria are responsible for producing energy in the form of ATP through a process called oxidative phosphorylation. However, in cancer cells, this process is often disrupted, leading to a shift towards glycolysis for energy production, known as the Warburg effect. This reliance on glycolysis, even in the presence of oxygen, is a hallmark of cancer cells and provides them with a growth advantage. Implications for cancer therapy. The role of mitochondria in cancer cell metabolism has important implications for cancer therapy. Targeting mitochondrial function has emerged as a potential strategy for treating cancer. By disrupting mitochondrial function or targeting specific metabolic pathways, such as fatty acid oxidation or glutamine metabolism, researchers aim to selectively kill cancer cells while sparing normal cells. This approach holds promise for developing more effective and less toxic treatments for cancer patients. Introduction to the Warburg effect. The Warburg effect is a phenomenon in which cancer cells exhibit altered metabolism compared to normal cells. Instead of relying on oxidative phosphorylation for energy production, cancer cells preferentially use glycolysis, even in the presence of oxygen. This metabolic shift allows cancer cells to rapidly generate ATP and other essential biomolecules needed for their uncontrolled growth and proliferation. The Warburg effect was first described by Otto Warburg in the 1920s, and it has since become a hallmark of cancer metabolism.